Thank you everyone for joining us this morning, this afternoon, depending on what time zone you are in. This is the 7th webinar for quantitative genetic tools for mapping trait variation to mechanisms, therapeutics, and intervention. And so this webinar series is sponsored by the NIDA Center of Excellence in Omics Systems Genetics and the Dictum. It was our way of compensating for the quarantine period, because usually we do a summer series or a summer workshop that covers some of these topics, but we've decided to put it online. And so my name is Laura Saba. I'm an associate professor at the University of Colorado Anschutz Medical Campus. And what the topic that I'm going to cover today is Introduction to Weighted Gene Co-Expression Network Analysis, so WGCNA. It's a tool that was described in about mid-2000s, around 2005, and it's been used by many people in the quantitative genetics field as a way to make co-expression networks, typically from RNA expression data. And so what I'm going to do today is go over, we'll talk about why we want to look at networks. We'll talk about basics of weighted gene co-expression network analysis from kind of a big overview of it, and then get into some of the WGCNA algorithmic details. So just a couple of housekeeping things. Rob and Shanak are going to help me monitor this chat, and so if you have questions, feel free to put them in the chat and we'll discuss it. The goal is to get through the webinar and all the slides within the first hour. And then with the last hour, we're hoping to have more of a discussion. So question and answers from everyone in the audience, not just me answering the questions. So going back to the beginning, I wanted to do a overview of what the webinar series is about to get you oriented if this is your first time attending one of our webinars. So the goal of this series was really to transverse this path from phenotype and genotype here in our analysis, to go from QTL analysis to genes and finally to that biological mechanisms. So we're taking it one step at a time and putting these tools together. We also post all of these webinars on YouTube, and so we will post that link shortly after it's done. And then the slides are available, and some other notes like issues that come up in the chat will be made available through our GitHub site and through our website of opar.io, so opar.io. So today we are strictly focusing on this end of that path when we're thinking about genes and getting to biological pathways and mechanisms for these candidate genes. So why networks? If you happen to come or hear my introduction to eQTLs, we did do a little talking about why networks, and we're going to reiterate some of that here. So why networks? Number one, we know that gene products do not act independently in the cell, and that most tissues are complex hierarchical networks. So there's a lot of communication not only within the cell, but between cells, across organs, across brain regions that are going on. And since this webinar series is sponsored by NIDA, one of the things to think about is for many diseases, including substance use disorders, they can often be conceptualized as a failure in network regulation. So we know that not a single gene causes a substance use disorder, but it's usually how these genes interact. And we also know that variants or in any one of the genes within the pathway could cause similar phenotypes or could cause similar symptoms. And finally, the generation of this network helps us to pull out a little more insight about our genes, and more than we could get by looking at only genes in isolation. But that was all kind of theory and big lofty ideas. But practically, what are the issues with independent gene analyses? So what happens when we're just doing an RNA expression study and simply looking for differential expression between our cases or controls or our exposed and non-exposed samples is that in the end, a lot of times we'll get hundreds of candidate genes from these independent tests, and we have to have some kind of strategy for deciding which genes to pursue further to take on to those functional studies. You know, one philosophy is to choose the most significant result using a p-value or an effect size or something like that. But as we all know, statistical significance doesn't necessarily indicate biological relevance. So it shouldn't be the only thing that we're using to decide on how to do our follow-up functional studies. Another approach that often happens is we take this list of hundreds of genes, we look at the top few and choose the one that's our favorite. We choose the one that we know the most about. We choose the one that we can find the literature about, which all of this makes sense in deciding what to pursue further, but it also biases some of our further functional studies to only those genes that are well annotated. And some of these genes that have little annotation, or some of these non-coding genes that we don't know what they do, tend to fall by the wayside. So even when we have a good candidate gene, there may be some challenges to thinking about what kind of functional validation you want to pursue. So like we talked about before, some transcripts may have little to no functional annotation or information, while some transcripts may play many roles, depending on what cell type we're talking about, depending on what environment we're talking about, both cellular and at the grander scale. And so the idea is that by using networks, we can help answer some of those questions. So we can get inferred biological function, so guilt by association type approach of a gene that's under-annotated based on other genes that are co-expressed with that gene. We can get the context in which the gene exerts its effect, too, by looking at genes within the network that have similar functions. So if we have a gene that's doing a lot in any cell and can take on many different roles, we can start narrowing down on the functionally relevant role by looking at what other pathways the genes that are highly associated are with. Finally, if one of our goals is to look for novel drug or therapeutic targets, by taking a pathway approach, we can think about not necessarily targeting the most well-known gene, but considering and looking for druggability of these targets. So not only can we build a drug to attack it, but also what kind of side effects may occur. And finally, near and dear to my heart is hopefully when we look at some of the more bulk style RNA expression studies, that by looking at similarity among genes within a module, we can start thinking about, for instance, in brain regions of interest or in any tissue, what kind of cell types may be responsible for the co-expression that we're seeing in our data. Like we've talked about before, there's many methods for defining networks of genes. So there's some classic databases out there that look strictly at protein-protein interactions and doing that in a very experimental and rigorous way. We know that there are several annotated pathways in gene ontology terms, such as KEGG pathways, Go ontology, Panther pathways, where people have spent hours upon hours curating all the information in our scientific publications to derive these pathways and put these genes together. And then finally, one of the other ways that we can define networks of genes, and what we're going to do today, is to talk about RNA co-expression. And so although we're going to talk about weighted gene co-expression network analysis today, please understand that this is only one method. It's not the end-all and be-all methods for identifying RNA co-expression. It tends to be a very good tool and popularly used because it's easy to understand, and it takes us one step beyond that, something like a k-means clustering. And I have Bayesian networks listed here. We'll talk about how our co-expression or WGCNA modules can eventually lead to something like Bayesian network when we do some follow-up studies in our candidate modules. So now we're going to just go through the very basic concepts of WGCNA and what makes it different than simply looking at correlation. So getting back to this idea of why are co-expression networks relevant is this assumption that co-expression as a measure of connection between genes. So in this graphic, what we've shown is the expression levels of 3 genes across different inbred strains, so different genetic backgrounds. And we see that gene A and gene B, as we go across different genetic backgrounds, their differences in expression are mirrored between A and B. So the concept here, or the theory here, is that gene A and gene B may be related to similar biological processes, since they tend to react to different environments. And genetic background here is our genetic environment. In a similar manner, then they most likely have some similar functional role. Whereas gene C does not have any kind of correlation or association between the other genes, so it most likely is not associated with the same biological pathway. And so obviously this is a theory, this isn't, again, the truth, but it's not going to cover every relationship that we'd ever see between 2 RNAs, so we may be wrong going in either direction. And so one of the first questions, at least from some statisticians or from my statistical point of view, is why not just use correlation? Why do we need something like weighted gene co-expression network analysis? And so there's 2 big differences between just simply looking at correlations and doing something like a hierarchical clustering. So the first one is that part of the analysis process in WGCNA is to transform this idea of a quantitative connection or quantitative association between 2 genes. So we're going to mathematically transform that to make our network mimic what a scale-free network would look like. And we're going to go into a little bit more detail in the next few slides about what I mean about a scale-free network. And then the other added benefit of weighted gene co-expression network analysis is that beyond just the scale-free network concept, we're also adding a more robust measure of co-expression. So now, instead of simply looking at the association between gene A and gene B, we're also going to take the approach of what are their indirect associations. So from a social network type perspective, are their friends with each other? And again, we'll talk just a little bit more about that in the next few slides. So if you're not familiar with a scale-free network, we typically think of a dichotomy of this over here is what we call a random network, and this one on this side is a scale-free network. And so the big difference here is in a random network, all of your nodes have about the same number of connections. So in our co-expression module, what this means is that most genes are connected to 3 or 4 other genes. In this graphic of the United States, what it looks like is sort of like the highway system across the US, where each major city has 2 or 3 major highways going in and out of it. Whereas a scale-free network, instead of every node looking similar, having a similar number of connections, what it turns out is that most nodes have very few connections, and only some nodes have a lot of connections. So the nodes that are down here with lots of connections, these we call hub genes. And so they play a critical role in the network in that they have many connections going in and out of them, whereas genes along here have just a few connections going in and out. And so our analogy here is the airline system, where we have just a few central hubs for some of our bigger airlines that we usually have to connect through if we're trying to get from one side of the country to the other side of the country. So biologically, there's several advantages to a scale-free network. And one of the first advantages is that it's very robust, so that if there's any failure in a node, it doesn't bring the whole system down. And we'll talk a little bit more about that coming up. And then finally, it's very efficient and adaptable, and it has what we call the small world property. So when thinking about this idea of scale-free networks being robust, if we look at these two graphics, I've done, this is my random network here. And when random nodes fail, you can see that it turns our network into these isolated nodes. And now, if we can't get in and out of some of these cities or some of these highways, it's really hard to get from the West Coast to the East Coast. Whereas in a scale-free network, if we randomly have any node failures, it's usually pretty easy to reroute our flights, right? So even if some of the nodes fail in this network, we can still get from the West Coast to the East Coast fairly easy. And the only way that we can really bring down a scale-free network is if we start attacking those hubs. Right? One of the pop culture references to the small world network is, if you've ever heard of the concept of, or the game of 7 degrees to Kevin Bacon. So and this reflects what we're talking about as efficiency, that we can get from any celebrity to Kevin Bacon within a few connections, right? So here we have Dwayne Johnson, The Rock, and Kevin Bacon, and they have never been in a movie together, but the concept of 6 degrees from Kevin Bacon is that we can easily find somebody that they're both connected to. So in this case, my example is Bill Murray. So they were both in movies with Bill Murray. So they were only 2 degrees away from Kevin Bacon. And it's just a description of how close-knit we are and how efficient it is to get from one point to another point in a scale-free network. And not to be outdone by The Rock and demonstrate some of these small world properties, although I am not a celebrity, I too have a path to Kevin Bacon in only 5 degrees. So what we see here is that you can connect me to my brother, who his college roommate was Jonathan Coachman, who was then on the WWE with The Rock, Bill Murray, and Kevin Bacon. So it really is a show of how small world properties work. Right? But it's not just celebrities that we think of working in this efficient manner, but it is also what we have observed in metabolic networks. And so this idea that to go from one metabolite to another, if we went along a linear array and had to go from 1 to 2, 2 to 3, 3 to 4 to get to our end product, it would be a really inefficient process, right? Instead, what we see in metabolic networks are these hub metabolites that it's easy to go from one hub to another. So instead of going through each one of these steps from 4 to 523, we can jump from hub to hub and get there much more efficiently and much more quickly. One of the additional rationale for thinking about a scale-free network and why it fits our data is this idea of selective pressure and evolution. So if we started off with a random network where the majority of nodes have the average number of connections and few nodes have many more or many less than average, then as our network grows, as we're randomly adding to each of these new nodes, the old nodes tend to build up the links, right? They get more and more links, where the newer nodes are going to tend to have fewer links. And so we call this preferential attachment and talk about the strong getting stronger. And so hopefully I laid out good rationale for this first step of the scale-free network. And now we're going to move on to what I mean by that robust measure of the network. So the idea here is that we want to see if the genes have similar friends. So if our idea is to look at the relationship between these two gray nodes, or these two genes in our case, on the right-hand side, for the low correlation, you can see that each one of my nodes is associated to a different gene, to 4 genes, but none of the genes that node A is associated with, node B is also associated with. So this has a low indirect correlations. On the other side, for this high indirect correlations, you see that both A and B are associated with 1, if you can see that. That's kind of poor color contrast. And both of them are also associated with 2. So it gives us more confidence that it is truly an association between A and B that we're seeing, and it's not something spurious if we can say that they're also both associated with these other genes. All right, so that kind of laid out our foundation about why we want to do WGCNA. And now we're going to go through in a little bit more detail about how we're going to do WGCNA. So, and I do, and forgive me for not saying this right off the bat, but I, of course, did not come up with WGCNA. Most of the data figures and everything that I'm showing you today, I've pulled out of articles by, Steve Horvath led this effort for many years. And so a lot of this information comes out of his lab, a lot of these figures, a lot of these concepts. And he, if you didn't happen to notice on the seminar announcement, there was a link to several really good papers. One of my favorites is the Weiss paper that it talks about good enough solutions. It does a fantastic job of laying out kind of the foundation for networks. And then there are a lot of tutorials to take you step by step through the WGCNA R package as well. So there's a link to that. So getting back to this overview of the WGCNA workflow, we're going to step through this one at a time. So we have construct a gene co-expression network, identify modules, relate them to external information, and then some interesting things to do after this analysis to really interrogate those modules a little further. So to be able to construct this gene co-expression network analysis, we have to do these 4 major steps. So we're going to start with the similarity matrix, transform it to an adjacency, transform it to a TOM, and transform that TOM to a dissimilarity matrix. And that seems like a little jargon when we first say it, but we'll take you step by step through those 4 steps and tell you how they relate to this idea of scale-free networks and direct and indirect associations. So first of all, when we calculate the similarity matrix, it's typically any way that we want to look at similarities between our genes. So a lot of times we will simply use this, a correlation coefficient, whether it's Spearman, Pearson, or a biweight, but it needs to be something that ranges from -1 to 1. And so that when we go from an expression data set that may have samples along the rows and genes along the columns, by taking the similarity matrix, what we get then is a symmetric matrix where it has rows and columns for each one of the genes in our networks. And each one of the cells in that matrix tell us the relationship or the similarity between those 2 genes. So the next step that we're going to do is to mathematically transform that similarity matrix to represent a scale-free network. So this is how that's incorporated into the WGCNA. And so one of our first steps before we can make that transition to the adjacency matrix is to decide whether or not we want to have a signed and unsigned or a signed hybrid network. And so what signed versus unsigned means is whether I want to consider 2 genes to be similar and in the end be in a co-expression model if they are negatively correlated. So that If I want to include those negative correlations as high similarities, then I'm going to do an unsigned network. If I want to make networks where all the genes are positively associated with each other, meaning if the expression of one goes up, the expression of another goes up, then I'm going to use a sign co-expression network. And this is really a matter of choice. My personal preference is always for the sign network. I think that a lot of times we can't do a tremendous job of predicting about what direction the relationship between two genes go. And when we think about pathways, there's always suppressors and activators in a pathway. And if we want to keep that information together, an unsigned way to go. Some of the argument for it is for a signed network is then you have a particular idea about the pathway being turned up or turned down, right? So once we've made a choice out of those three, we need to transform our data. And to do that, we need to decide on what's called a soft threshold beta. So it's a certain parameter in this transformation that's going to be unique to the data set that we need to evaluate to see for our particular data set, what value of that beta do we need to get it to resemble a scale-free network? And so how do we mimic that? So if you remember, these were our graphics from before that was looking at the distribution of number of links between number of nodes, right? Or it's looking at the distribution of the number of links per node. And so in our scale-free, remember we had lots of nodes with few hits and only a few nodes with lots of hits. And so this black line represents the our measure of connectivity. So just a sum of all the correlation coefficients for that gene. Looks rather like a random network when we first see those correlation coefficients. We see kind of a general bump kind of in the middle where most people have kind of a similar connectivity. So what we do mathematically is we take those correlation coefficients or whatever we're using in our similarity matrix and we raise them to a power. And so what that does, and you can see in this graphic, is slowly shift that distribution to the left. And so in the end, when we get to 6 or 7, we're looking much more like the scale-free network over here and less like the random network. And so all we're using is a mathematical transformation. How do we evaluate what kind of soft threshold to use? So within the WGCNA R package, what they're doing is evaluating many soft thresholds and then looking at the R squared for how well it fits a scale-free network. How close are we to a scale-free network? So a higher value means it's more similar to a scale-free network. And then the other thing that we evaluate is the mean number of connections per gene. And so we remember we want that mean number of connections to go down because most genes won't have that many connections. And so in general, recommendation is an R squared of 0.8 or above. And then also for when we see kind of an elbow in this distribution. Oops. Sorry about that. So when we start to see this elbow. So in this demo, we're really looking at that 6, 7, 8 region is where we first cross that threshold of 0.085 and when we first start seeing that elbow in our soft threshold. So once we've decided on that soft threshold, we actually apply it to our data set like we talked about. So we take our similarity matrix and raise it to the power that we've chose. So raise it to that soft threshold beta. Then the second part, so now we've gone from similarity to adjacency, and now we're moving on to what we call a topological overlap matrix, or TOM for short. And that's when we start incorporating those indirect associations. And so this is the nitty-gritty details on the statistical formula for that. And so I'm going to break it down a little bit here. So this A1ij, if we're looking at the Tom value between gene I and gene J, then the IJ is the direct relationship. And LIJ is the indirect relationship between the two. And then this is just a standardizing or scaling factor down here, this combination. So our indirect, what we've done here is multiply the correlation or the adjacency between gene I and some other gene and gene J and some other gene. So when both are correlated with a gene, that makes that number bigger, right? Where if I have it where one has a high and one has a low, that's going to dramatically reduce that number. And so what we're looking here is for concordance of their association with other genes. And again, it's super simple to calculate in the R package, to calculate that Tom. So to give you just a glimpse of what we're going to do with these network measurements is we're going to do some hierarchical clustering a little bit later on. And so for that step, we really need a distance rather than a similarity. So the last step in this creating a network is to simply transform it from a similarity matrix into a dissimilarity matrix by taking 1 minus the value of the Tom. All right, so now we have a nice big matrix that describes the relationship between every gene and every other gene. Now what we want to do is use those relationships to identify modules of co-expressed genes. So one of the basic ways for doing this is what we call hierarchical clustering. And what that does is, for instance, it takes these 6 values, it looks for the distance between each pair of these 6 nodes, so 6 genes in the case of a co-expression network, and it clusters together the pair with the smallest distance. So if in this case, this would be our first step is to cluster the A, the B, and the C. And so once it does that first cluster, then it's going to reevaluate the distances. And so instead of now B being associated with each one individually, we're going to look at the distance between B, C, and A, B, C, and D, B, C, and E. And we're going to keep doing this iterative process where we evaluate all the distances between our nodes or our merged nodes until we get to a point where we've gotten all our nodes to merge together. And so this is what we mean when we talk about hierarchical clustering. Oftentimes we'll see these results displayed in a dendrogram. And so in a dendrogram, we often see it, this along the y-axis is our distance at merge. So for instance, 0.1 and 0.4, they only have a distance of 1. And so that's where the line was added and that's where they were merged. Whereas 6 and 7 were merged at about 1.5, right? And so on and so forth until we all get clustered together. When we're just using traditional hierarchical clustering to identify co-expressed modules, what we often do is just set a cut height, where it would be a vertical height within our graph. We cut it across, and how the items clutter at that cut height, it's the clusters that they form. So if my cut height was there, 3, 5 would be a cluster, 6, 7 would be a cluster, 2 would be by itself, and 1, 4 would be a cluster. Right. WGCNA by default has a much more sophisticated and iterative process for identifying clusters rather than just simply taking a vertical height. And so this is a whole area of study that they've looked into about how to do this in an iterative process so that they can identify nested clusters, that they can be flexible, and they can combine both the advantages of hierarchical clustering and partitioning around medioids, giving it better detection of outliers and things like that. And so there's for the details here, this could be a whole presentation in itself. I would refer you to the Langfelder and Horvath paper that outlines it in great detail. And within this paper, they show how this particular type of tree cutting does really well on capturing some of the information in the data. So the data was simulated, but based on this first row, and then these are the different results across the different types of clustering methods, including the dynamic tree cut and the dynamic hybrid tree cut that do well at mimicking what was simulated. Just like all the other functions, there is R code to do this dynamic tree cut. They ask you a couple, I've just shown a few options here for the dynamic tree cut. There's many, many more, but a couple things that I want to point out that we found very useful in our lab is this idea of deep split. This is a number between 0 and 4. The higher the number, the smaller the modules that you're going to get. Right, and so if you're interested in the smaller modules and breaking some of the bigger modules down, then you use a deep split 4. If you're more interested in the biological implications of some of the bigger modules and want to be able to do some of the downstream analyses that require larger numbers of genes, then you'd want to do a deep split closer to 0. And the other thing that's controversial, not controversial, but there's lots of opinions in the field, is this final parameter about a minimum cluster size. And so I have written down 5, which means that if a cluster has less than 5 genes, then they're not going to call it a module. The default is 20. I've also heard lots of people say 30. And it really depends on what you're going to use these modules for and how you want to interpret them on whether or not you're interested in modules that go down to a size of just 5 genes or not. And so I showed you step by step the R code into generating both this network and identifying modules. But the group with the WGCNA package have done a fantastic job of making it super easy to implement this analysis and have put a wrapper around all of those functions, if you're familiar with some of the terminology in R, so that we actually only have to do a 1 line of code to get from our raw data to our modules. And so the next step is, how do we relate modules to external information? We have several different options, and the 3 that I'm going to talk briefly about today are we can associate these modules directly to external traits that are quantitative through module eigengenes. We can also test every one of the genes in the module for their association with a quantitative trait and then aggregate those results rather than aggregating before the association. And then finally, to get an idea of function of the module, we can use some of our enrichment techniques to get at what known functions are there some known functions of genes in that module that we can start to infer about what biological processes, what tissues, what cell types does this module represent. And so some of the typical things that we can associate it with, we can go with DNA variants. We talked a little bit about this during the introduction to EQTL webinar. We can associate it with lots of different omics types of data. We can do behavioral and phenotypic data, the functional information that we talked about. So again, thinking about KEGG and GO and databases like that. And then finally, we can also look for tissue and cell type specific markers within our co-expression module to get an idea of relevant areas. So if we're looking at whole brain, or is there some relevant regions? Are there some relevant cell types to this particular association? Again, this is a little bit of a repeat from before, but module eigengene is this concept of a way to summarize the expression patterns across our samples for a given module. So instead of having a database or a matrix that has 40 genes across 18 samples, how can we summarize that into one value per sample to get a general idea of this pattern that we're seeing, for instance, in this graphic over our samples? And so all we do, the WGCNA algorithm includes what we, first principal component is what it's calling its module eigengene. So that's from a principal component analysis. We can examine what proportion of variance of the total across all the genes is explained by that PCA to get, or for that first principal component to get a good idea of how representative it is for that module and whether or not taking it on to further analysis is a good idea. And it gives us something easy to work with, right? So it reduces that multiple testing because now we're just looking at one module eigengene against our trait rather than associating every single gene in that module with our trait. For linking with DNA variants, we talked a little bit about my module eigengene QTL. We can do a QTL analysis with the module eigengene, just like we could do a behavioral or phenotypic QTL to look at where in the genome is this eigengene associated with. We can also, this is kind of a messy figure that I pulled out of some of their tutorial information, but we can, one of the big benefits of the WGCNA R package, I'll have to say, is that some of the graphics that they do are very creative. And what they've done here is these are all modules for their network. And they've looked at their association across all of these phenotypes, and they've color coded those associations. So this is the correlation with the between the module eigengene and the phenotype. So we can start to see patterns across similar traits. So here we kind of see this cluster around the traits that are associated with fat, so total fat, other fat, abdominal fat. We can also get some really strong hits for some of these modules. And the other benefit is we can see if a module is associated with more than one phenotype to get us a little bit more detailed information. I just glanced at the chat and saw one of the comments that I want to say it while we're here. Although they do this figure as an association, they do red for positive association and green for negative association. That direction of association is only interpretable if you did a signed network, right? So if you did a signed network, then a positive association would indicate increased expression is associated with increased values for that phenotype. But when we do a unsigned network, the directionality has very little meaning, and it's just comes up by chance, because you'll remember with an unsigned network, some genes in the module may be positively associated with a trait while other genes are negatively associated with a trait. We can also aggregate over gene level association. So for some instance, maybe you have to do it at the gene level and you can't do it at the module eigengene level. What we can do is look at all the genes, their individual association with the trait, and the example approach that they use in the tutorial is simply to take the average. But you can see how we could easily do something like a meta-analysis p-value or something similar like that. The final way of linking external information to the module that we're going to talk about today is enrichment analysis. And so the goal of enrichment analysis is to determine if the number of genes within the module that are associated with a specific pathway, ontology, cell type, whatever your category is more than expected by chance. And so in this demo graphic that Sheree Wood put together, you can see she plotted both the observed and the expected number of genes. So this would be number of genes along this axis. And you can see for these two data sets, or for these two ontologies, there isn't much difference between what we would expect simply by chance ending in that module, depending on the module size. But within the wound healing example, we see many more genes than we would expect by chance. And so we can use standard statistical methods like a Fisher's exact test to get a p-value to really give us a statistical inference for this association. Just to remind you and to give you a couple resources for functional enrichment. EnrichR is one of my favorites. It's a web-based tool. It looks at over 160 different libraries. So not just KIG pathways and gene ontologies. It's super simple to use if you've ever used it. The only drawback that I see to it is that it doesn't allow you to set a background data set. So if you want your background data set to only be genes that are expressed in brain, for instance, or ones that you have tested, it's not going to allow you to do that. The Panther database, which has the most up-to-date gene ontology or GO information, is another one that's out there that's web-based. And then finally, if you like to incorporate it into our programs, there are API tools for Ensemble and KIG. Ensemble even has the BioMart library package that it's really easy to bring that data directly into R and calculate those statistics by yourself. You can also do enrichment for tissues, brain regions, and cell types. So EnrichR has some of that information, but there's also several lists that are already in the WGCNA package that can be used to look for functional enrichments that they have put together. Just because I'd been working on some single cell data the week before I did this, there's also several databases that have cell-specific markers that we could use in these types of analyses. And these databases tend to just keep growing and improving with the more single cell RNA-seq that's being used. We can, moving on, we can study relationships between modules. And so here, I'm just going to touch on this briefly, because again, this is a really in-depth concept about how not only do we look how genes cluster within modules, but looking how modules are related to one another. And so this is a whole field. We call this Eigengene networks. So we can think about a bigger picture there. And then just to give you a demo of how it's been used, at least in our hands, this is a publication that Lauren van der Linden, who I think I saw log on, she did with my lab, where she was looking at Eigengene networks across brain regions. And she used some of Mike's data, I know Mike's on here too, to look at how these modules relate with one another and relate to the whole brain data. The last category before we finish up here are how do we find key drivers in interesting modules? So now we've gone through, we've identified our modules, we've said this module is associated with our trait, whether our trait is alcohol consumption or our trait is blood pressure or any of those traits. We now want to look at that module in a little bit more depth. And so looking at the genes within it and starting to think about which ones we could target for functional validation and which ones, how we want to go about that validation. So one of the first things we can do is we look at intramodule connectivity. So how connected is the gene to all other genes within this module? And this could be calculated as simply the sum of all pairwise correlations of a gene with all the other genes in the module. It's a term often used, whether you use the similarity matrix or the adjacency matrix or the Tom matrix is really up to you to calculate this intramodule connectivity. But what we typically say is that the gene that has the highest intramodule connectivity, we tend to call that the hub gene. Sometimes for our bigger modules, we may identify multiple hub genes. So these genes are at the heart of the network. And so there is some controversy on the field. I've seen talks go either way about whether we automatically take the hub gene and target that, or if we want to go with some other genes on the periphery and what would be the best candidate for therapeutics and things like that. We can also do an assessment of module cohesion related to an individual gene. And to do a little self-promotion, we recently published a paper where we use this idea of module cohesion to decide on which gene within our co-expression module to target for knockdown and genetic manipulation. And so the question here is whether or not we can identify what gene is responsible for a lot of the co-expression within the module. And so for this analysis, we're relying on this idea of partial correlation as a tool for causal inference. And so the theory is that if changes in gene A cause changes in gene B, then regardless of the source of the biological variation in A, it should correspond to variation in gene B. So whether or not it comes from a cis-QTL or other trans-QTL, however I change gene A, this should result in a change in gene B, if this truly is a causal relationship. And this is a good intro and teaser for the next few webinar series. We're going to do a lot more talking about in more detail some of this idea about causal inference, and especially in genetic studies and omics studies. And so how partial correlation tests this is if we're looking at the association between two genes, right? And let's say gene A here along my x-axis has a strong cis-EQTL. So we see this pattern of really high values in people with genotype A and really low values in people with genotype B. If gene B is also associated with that gene, then it looks like when we do a correlation that these two genes are correlated with one another. What a partial correlation does is go in and say, okay, but within these groups, these dichotomized groups, are they correlated as well? So do we still see correlation even when that source of variation is not a cis-EQTL? And so what we did was we looked at our original co-expression network structure and looked at the relationship between genes when we controlled for different elements. So when we controlled for the module eigengenes, so the SNP that was contributing to the variation in this module, we saw our connections go down. But when we controlled for the expression of our candidate gene, which in this case is LRAP, we saw that a lot of the associations that we were seeing disappeared in our network. And so the hypothesis here is that LRAP may be driving a lot of the co-expression that we're seeing in this network. And then the final point on going a little bit further, and again, like we said, we're going to go into much more detail in further webinars, is that now that we have kind of a smaller, more manageable set of genes, we can start thinking about using Bayesian networks to direct some of those edges. And especially when we can hook that SNP there, we can be able to gain a lot of information. Again, I'm not going to go into detail because these are a whole other webinar by itself. But Brian Lusk, a grad student in my lab, has used these techniques successfully when he was looking at a co-expression network associated with alcohol metabolism. So he got this module from our tools with WGCNA and then was able to transfer it or translate it into a Bayesian model based on the narrowing down of these genes and getting the SNP and phenotype information that actually gives us a lot more information about how this SNP is influencing both circulating acetate levels and alcohol clearance. So finally, I'd be remiss if I did this presentation and didn't tell you about some of the limitations of WGCNA. Like I said, there's no perfect co-expression network analysis tool. There's different tools are better for different reasons. And some of the things that we have to keep in mind when using a tool like WGCNA is, first of all, we're assuming linear or even just monotonic relationships among genes, right? So we're not looking at anything like both high and low levels of this gene cause mediocre levels of that gene, right? Scale-free networks, although we made a big, we have several lines of evidence to think that that's how protein coding genes interact. We really don't know much about relationships or what kind of networks we would expect when we start throwing in non-coding transcripts. So not just relationships between two non-coding transcripts, but how they relate to these protein coding transcripts. And even beyond that, to other omics data. There is some work on robustness and reproducibility of these modules, and I refer to a paper, again, by Lingfelder and Horvath that has some tools on how to judge whether or not your particular model has been preserved or reproducible. And so in the end, from the methods that they propose, you get an overall value for your network. And sometimes we actually want to know more about specific connections within the network. What we've found very anecdotally in our studies is that the most highly connected genes are usually most robust. The ones that are less connected when using that intramodule connectivity measure tend to be the ones that are a lot less robust and can jump from one module to another module by making simple changes or redoing the analysis. One of the big concerns, especially with rodent models, where there are long stretches of linkage disequilibrium, is that sometimes our co-expression is due to the fact that both genes are near each other and associated with different SNPs that are in linkage disequilibrium rather than representing a true biological function similarity. And then finally, both a benefit and a disadvantage of co-expression analysis is that we don't include any prior information. So we don't include some things that we already know into the models to help make those models a little stronger. And so that wraps up this broad overview of WGCNA. So just to recap quickly, why networks? It gives us more context. It lets us think about functional hypotheses. And we do believe that it more closely mimics what's really going on in the cell and the communication between tissues. We talked about the two differences between WGCNA and simple correlation is that scale-free network assumption and indirect and direct associations. And then finally, we tried to break it down into steps to make it understandable on how the algorithm works, but knowing that a lot of these things can be easily done in one step using some of the WGCNA tools. So just to reiterate, this was sponsored by the NIDA Core Center of Excellence in Omics and Systems Genetics that we're trying to do as a resource center. We're trying to provide tools and training and things that will be useful to the community that will promote this idea of looking at systems genetics and looking at genetics in general when thinking about behaviors and addiction research. And then finally, I'd like to thank the members in my lab. A lot of this work is actually their work, things that they've put together and I've been able to put up on this slide. And so they've been extremely helpful. And I also get some financial support from a resource grant from NIDA. That's the heritable transcriptome in alcoholism. All right, so I think that wraps it up for the formal presentation. We'd now like to open it up to questions.
Thank you, Laura.
Yes.
I think in the chat there have been a few questions. And I think the first one is, what's your Erdos number?
I don't know that. sorry to be a disappointment. I only know my degrees to Kevin Bacon. I guess I need to get my priorities straight. But I see the next one from Mike Miles. So there's a few different things. So the first one is normalization of RNA-seq data for WGCNA. I can go ahead and give my opinion on that. You know, there's a couple options out there. You can do something like a regularized log transformation, a variant stabilizing transformation, which gets that RNA-seq data to look at least a little bit more normal. And then you can use tools like a Pearson correlation, or you can go straight from something like a TPM, but use a non-parametric correlation. So that's one of the benefits of that first step for calculating that similarity matrix, is that you can use a method that's appropriate for your data, that you get to choose whether or not your input data is appropriate for a linear type parametric test, or if you're better off using a rank-based test like Spearman for it. So I'm always a big fan of the transformation and then looking at both the Pearson and a Spearman correlation with that one. Does anybody else want to chime in on that answer?
Laura, one, nice talk, nice presentation. This is Mike. Well, I've seen a number of papers and gone back and forth with it using VUM as a variant stabilizing transformation for RNA-seq data specifically. Have you used that at all? It's I guess it's within, is it within Lima or Combat?
Yeah, I can't remember where it's in, but yeah, so we've tried Loom before, or VUM before, and what our experience has been is that the VST or the R log works a lot better than VUM. We see it just wasn't behaving well in the data sets that we were using it in. So we didn't do a formal simulation or anything like that, but in our anecdotal experience, it did poorly.
Okay, thanks.
So, and I think the other two are just as easy to implement. And I tend to see the other two more in publications than I do the VUM here lately, at least.
What about the issue, and this gets kicked around or a lot in various blogs, et cetera, about the selection of genes before you even start your analysis in WGCNA? Some people pick the top 5,000 most variable genes. There's just a whole bunch of strategies about how you choose which genes to input into WGCNA rather than using all of them, for example. What are your thoughts there?
So again, my opinion, I haven't done a formal study on it, but I put all genes in that are expressed at detectable levels. And the reason why is that I've made that scale-free assumption. So as soon as I start only picking the variable genes, then I'm starting to get away from some of the biological rationalization for that scale-free network. Whether or not that has some true statistical consequences or not, I tend to put them all in and see what happens.
Right. We usually do a cutoff for the expression level, which is, again, somewhat arbitrary. And then we use the mean, I guess it's the median absolute deviation and plot out the variance of the expression and trim off the bottom 10% of genes or something like that. But it's just, we haven't validated it totally either. So, but I think that was something that Horvath recommended a few years back, but I was just curious if you had new thoughts on it. So.
Yeah. You know, and one of the other reasons that, you know, it might not impact kind of the way that we apply it in the lab is that we're really looking for some of the smaller modules. And so what we see tends to happen is then that those genes that weren't variable tend to get thrown into that first module that's really big, right? And there's very little variation in that one. So it does probably depend on how you're gonna interpret it or what you're gonna focus on in the end.
Right. Oh, one last thing that I mentioned there, which I think is really important for people to think about, because there's, you know, you see a lot of variation in this in the publications using WGCNA, is the actual number of samples that are used in the analysis. And I know that Bob Hitzman and you know, and others in Oregon have done some evaluation of that and think that around 30 independent samples are sort of a good number to get robust networks. But Horvath and colleagues have mentioned smaller numbers than that. So do you have any guidelines there for to mention to people?
Yeah, I typically think that anything below 20 is suspect. 30 is optimal, right? So if I was planning a study of inbred strains for a grant application, I would say that 30 is the minimum, right? And so what we've also seen is that sample size also is going to depend on what your study design is, right? So when we're talking about inbred strains and we're talking about strain means that we're using to get our number of 30, right? 30 strains, we're going to need fewer of those than somebody who's doing like an HS study, right? Who has a lot more variation. And again, it's because our study design is looking for co-expression that is due to genetic background, because that's the only thing that's varying in our population. And so when we're looking for a network that varies due to differences in genetics, by having an inbred strain and measuring 3 biological replicates and then taking the mean, we've really eliminated some of that extraneous variation, right? That we don't necessarily care about and want to reduce in our final model. Whereas if you're thinking about an analysis across time with many treatments, you might be thinking it might be a different sample size for that, right? So, you know, we've found, and the other thing that we have found, like we talked about before, is that even with a smaller sample size, what I've seen happen is that if you have a module of 15 genes, 7 will be recovered the next time. So the connections between 7 of the 15 are really robust, but the entire 15 is not robust, right? And so in my mind, the differences in sample size, the higher you go, the bigger the proportion of genes within your module that are going to be robust. Right? Does that make sense?
Yeah, no, thanks. It's good discussion.
I think that cover all of your questions, Mike?
Yeah, no, I thought they just were maybe good things for everybody to, because those are some major issues that people have to face when they start using WGCNA, and are good to think at before you start going through the pipeline. So, yeah.
Right.
Thanks.
And the other thing is, I've had people ask, I mean, I'm fairly focused on most of my research on rodent models, but I know especially in making that transition from rodent to human, that the human data needs many more samples to get robust modules than what we need in our rodent models, which is probably what we would expect as well. So Rob mentioned that the eigengene and the directionality in the eigengene. And so, yeah, just to reiterate, in an unsigned network, associations between an eigengene and a trait, I wouldn't read anything into the direction of that association. I would just think about the magnitude of that association. And then, yes, I am remiss. There's several good geo-ontology or genotype enrichment analyses out there. So Web Gestalt is a good one. And then, of course, Gene Weaver, which is, you know, I missed it because it does so much more than just functional enrichment. And the fourth Friday in October, Alyssa or someone from her group will be doing a detailed webinar on Gene Weaver and give us a lot more details about that.
Laura, on the sign issue, even if it was an unsigned network, isn't the sort of the construction of the eigen trait sort of arbitrary in sign? Because, you know, anything can be, like when you do principal components, which is what the eigengene is, the sign of that eigenvector can be flipped without any change in the performance of the trait. So wouldn't that imply that the sign of the eigentrait is arbitrary, whether it's signed network or not?
Yeah, I mean, I see your point. I guess my thought was that with positive associations, you'll tend to get positive weights, but that's not true. So I see your point. So yeah.
Yeah, I think that's right. That you really can't depend upon the direction of the correlation between your eigen gene and your trait, even with signed networks. So you have to really check it out.
I think that though the idea behind the heat map that we looked at, that was trait versus module eigengene, is more not necessarily whether it's red or green, or it's more of, oh, both of these traits had a red association. So they had association in the same direction. So those traits are probably positively associated with one another as well. Maybe a way to why you would keep the red and green in that particular heat map, at least for visualization purposes.
Right. I think that's a good point. I think you're safe there. Yeah.
Mm-hmm.
There's another question from Nicholas Waddell.
Okay.
At the bottom.
Yeah. So it says, I work in a rodent models and our lab uses drug self-administration and other behavioral measurements. My question is, like RNA-seq data, do behavioral measurements need to be normalized or transformed? So again, it gives you a little bit of freedom once you have that module eigengene. If you're going to use a Pearson correlation between the module eigengene and your trait, then your trait needs to be normally distributed. But the beauty in like doing it through a module eigengene is that you can really do any kind of association you want. You could do a more complex linear model with cofactors that you adjust for. You could do non-parametric tests, anything you want to do. So whether or not you have to normalize your behavioral data is going to depend on what kind of test you're using for that association with the module eigengene. We can assume that our module eigengenes are relatively normally distributed. It's just whether or not the other one is. Just to reiterate on a couple other, interesting ways that people have been using WGCNA. People have looked at splicing through WGCNA and networks like that. They've looked at other omics data like proteomics and metabolomics through this data. I think that one of the reasons that WGCNA is as popular as it is is not that it's an overly complex model, but if you're familiar with R, they've done a fantastic job of putting a package together. And so one of my general teaching philosophies is that WGCNA is a really good method to start off with when learning about network analyses. It's a little bit more clear and less statistically complex than other methods. And the tutorials that this group have put together are fantastic and easy to follow along and in depth. And so I definitely think it's a good one to start with, and then think about extensions or, you know, Bayesian networks and things like that as a second step.
Laura, this is Mike. I'll just add one little comment here. As one of the favorite things that we like to do within WGCNA when we do some of these analyses is with a given module to try and highlight the importance maybe or our interest in that module's correlation with a given phenotype. Is the plot within WGCNA where you plot on, I can't remember what it's called, but you plot basically the gene-gene correlations on one axis and then the gene phenotype correlations on the other axis for a given module. And so what happens is that the genes that wind up at the one, if you see a strong linear relationship, then you're seeing that basically your connectivity of your network is highly correlated with the trait, and that the genes that are more connected have a stronger correlation with the trait. And that really helps you sometimes focus in on genes that are really of biological interest. And at least for us, it seems to make a graphical picture of, oh, this is a network we really want to pay some more attention to. So I'll just give that a little toast. So.
Yeah, and the other thing that it's interesting too, another validating point of that graphic is, again, going back to what's robust and what's not robust in the module. So the lower the connectivity, the less robust. So it's somewhat reassuring to see that the lower ones are the ones that are less connected to the phenotype or less correlated with the phenotype when moving forward.